Today we are going to start now the lecture that is a genetic counseling. In last period we were studied that is a disease, and what is a genetic disease, chromosomal disorder. Disease are the condition of the body when the body organs do not function properly, that is a disease. And then syndromes, a set of disorder called a syndrome. So there are mainly the different kinds of the diseases, so out of which genetic and chromosomal disorder. uh, Generally, in last period, you were studied very well, that is a Down syndrome. That is a Down syndrome, that is a trisomy of 21st chromosome. And in case of the maternal age is very high, so there will be the more chances for the that babies born, so that are suffering from the Down syndrome. So in case of during the there is a some married couples have some, there is a in case of problems, so doctor suggests to them to find out all the test of that developing baby, and give the suggestion to them. So that type of suggestion, that is a genetic counseling. And in today's session we start now the genetic counseling. So the genetic counseling is advice based on the analysis of the probability that patients have a genetic defect, or of the risk that prospective parents may produce a child with genetic defect. In the latter case, genetic counseling involves the presenting the available options for avoiding or minimizing those risks. If a serious genetic defect is identified in the fetus, one option is abortion, means pregnancy termination, medical termination of pregnancy, MTP. Genetic counseling gives people an understanding of the genetic problems that are or may be in their families or prospective families. Genetic counseling includes a wide range of information on human heredity. In many instances, the risk of having child with genetic condition maybe stand in terms of the probabilities. In other, where the role of heredity is not completely clear, the risk is only estimated generally. It is the responsibility of the genetic counselor to give their clients clear, unemotional, and broad statements based on the family history and on their knowledge of all relevant scientific information and the probable risk of giving birth to child with genetic defect or any kind of chromosomal abnormality. Genetic counseling generally starts with pedigree analysis, means evolutionary history of living organism, that is a pedigree, and obtain their record called a pedigree record. So this can be find out with pedigree analysis at the study of family tree, and careful record maintained, that is the phenotypic and the genotypic. Phenotypic record can be observed by externally visible characters, and genotypic record obtained by the genetic constitution of both families over number of generation. Pedigree analysis is used to determine the likelihood that a particular allele is present in either individual heterozygous or recessive mutations, or by fetal analysis. Assay for the enzyme activities or protein amounts are limited to genetic disease in which the biochemical condition is expressed in the parents or developing fetus. Carrier detection and fetal analysis so mainly inform parents of the risk and the probabilities of having child with mutation. What is a mutation? So all of you know that sudden accidental inheritable changes takes place in the genetic makeup of organism, that is a mutation. Then carrier detection identifies the people who are heterozygous, means two type of genes are present in one pair. Same type of gene are homozygous condition, and same type of gene, and the two different kind of genes are present, one are dominant and other are recessive, that is a heterozygous condition. And mostly the heterozygous condition are the carrier, because one genes are dominant and other are recessive. So dominant gene hide the expression of the recessive gene, so that person or that patient not to be totally normal, but it will be carrier for that gene mutation. The heterozygous carrier of the mutant gene usually is normal in phenotype. if homozygosity for the mutation result in serious deleterious effect. But when the genetic makeup are homozygous condition, both the genes are similar, so that person is affected or patient, that not to be the carrier. There is a great value in determining whether the two people who are thinking of having child and are both carrier in that situation, they have 1/4 chance of having child with that trait. Carrier detection can be used in cases in which a gene product, that is a protein or enzyme, can be analyzed, because all of you know that one gene one enzyme hypothesis. Genes are the fragments of DNA, and that genes or DNA give rise to RNA by the process of transcription, and RNA give rise to protein by the translation, and protein give rise to enzyme, because all enzymes are protein in nature. The heterozygous is expected to have approximately half the enzyme activity or protein amount as a homozygous normal individual. So, fetal analysis. So, second important factor of genetic counseling is find out whether fetus is normal. So, out of them, doctor suggests one, perform a technique, that is amniocentesis. What is this amniocentesis? For this purpose, here amniotic fluid is withdrawn. Amniotic fluid is injected. A needle, specific hypodermic needle is injected in the uterus of the pregnant lady, and that with the help of that needle remove the amniotic fluid which are present in the uterus. Amnion are the extra embryonic membranes. There are the four kinds of the membranes, such as amnion, chorion, yolk sac, and allantois. So amnion secretes the amniotic fluid, and this amniotic fluid provide protection to that developing embryo. It acts as a cushion and absorb the shock and jerks of the embryo. Shock and jerks, it protect the embryo from any kind of mechanical injury. So in during this process, the second important aspect here, the amniocentesis is one in which the fetus can be done. A fetus develop in the amniotic sac, amniotic fluid surround it, serving as a cushion against shock. Amniocentesis is a syringe needle is carefully inserted through the mother's uterine wall and into the amniotic sac, and the sample of amniotic fluid is taken. The fluid contains cells that the fetal cell, or that fluid contains the fetal skins has slug up. This cell can be cultured in the laboratory and thus examined for protein of enzymatic alterations or any kind of deficiency. DNA changes and the chromosomal available DNA changes and any kind of chromosomal abnormalities can be checked by this technique. Amniocentesis is possible at any stage of pregnancy, but the small quantity of available and the risk of the fetus makes it impractical to perform the procedure before the 12 week of gestation period, because the gestation period means the pregnancy period. So because the amniocentesis is complicated and costly, it is used primarily in high risk cases such as mothers is over 35. Another method for fetal analysis is a chronic villus sampling. This chronic villus sampling, here the procedure is done between the 8 to 12 week of pregnancy, earlier than for the amniocentesis. The chorion is a membrane layer surrounding the fetus and consisting entirely of the embryonic tissue. So chorionic villus tissue sample may be taken for the developing placenta through the abdomen. Placenta, it is a temporary connection between the extra embryonic membranes of the fetus and the maternal uterine wall. So during this second technique, that is a chorionic villus sampling, chorionic villus sampling, there is a chorion. These are the extra embryonic membranes. So to remove the fluid from that chorion, and to check, just like the amniocentesis, in case of the amniocentesis, amniotic fluid is withdrawn. But in case of this chorionic villus sampling, chorion is a chorionic villus tissue sample may be taken from the developing placenta through the abdomen. And why the vagina, using biopsy forceps or flexible catheter added by the ultrasound. Once the tissue sample is obtained, the analysis is similar to that used in the amniocentesis. Advantages of the technique are that the time testing permits the parent to learn whether the fetus has genetic defect earlier in the pregnancy, than with amniocentesis, and that it is not necessary to culture the cells to obtain enough to the biochemical assay. So fetal death and the inaccurate diagnosis caused by the presence of a maternal cell are more common in chorionic villus sampling than in the amniocentesis, and this is the one of the drawbacks of this technique. So students, here finish this topic, that is the amniocentesis, and next lecture we will study next topic or next genetic and chromosomal disorder. So thank you very much.